The rising popularity of NR, nicotinamide riboside, and NMN, nicotinamide mononucleotide, in the longevity space has brought attention to the fundamental role of NAD in aging and cellular health. These compounds, often presented as cutting-edge solutions for reversing or slowing aging, have gained traction largely through preclinical studies showing enhanced mitochondrial function, improved DNA repair, and increased sirtuin activity in model organisms. While their promise is real and supported by sound biochemistry, it is essential to recognize that much of the enthusiasm surrounding NR and NMN is speculative, hinging on limited human trials and commercial narratives. In contrast, niacin, despite being less glamorous, is a clinically grounded molecule with a rich therapeutic history, regulatory approval, and evidence of anti-aging relevance that has been overshadowed by the flashier newcomers. Niacin's role in human health has been documented for over half a century. It has long been used to manage dyslipidemia, reduce cardiovascular risk, and support brain function. While it was not originally framed in the context of anti-aging, its clinical impact intersects with many hallmarks of aging, chronic inflammation, mitochondrial dysfunction, and metabolic decline. Unlike NR and NMN, whose benefits in humans remain preliminary, niacin's ability to influence these aging-associated processes is well supported in clinical literature. Its efficacy in lowering LDL cholesterol, raising HDL cholesterol, and reducing triglycerides contributes directly to cardiovascular health, which remains one of the most significant age-related health concerns worldwide. In addition, niacin activates GPR109A, also called HCA2, a receptor with anti-inflammatory properties. GPR109A signaling reduces pro-inflammatory cytokine production in immune cells, contributing to healthier immune aging and metabolic stability. While NR and NMN may indirectly reduce inflammation through increased NAD and subsequent sirtuin activation, niacin's pathway is more direct and clinically observable. The GPR109A activation, though responsible for the characteristic flush, is mechanistically meaningful. It's a pharmacodynamic marker of anti-inflammatory engagement, not a mere side effect. On the other hand, NR and NMN research in humans is still developing. While early trials have shown that both compounds can elevate NAD levels in blood and potentially muscle tissue, the translation of those increases into tangible health benefits is uncertain. For instance, several randomized controlled trials have shown that while NR can increase NAD and modestly improve some biomarkers of metabolic health, it has not consistently improved glucose tolerance, insulin sensitivity, or cognitive performance in healthy adults. Similarly, NMN has shown promise in small trials involving older adults, including modest improvements in insulin sensitivity or muscle performance, but these studies are often limited in size, duration, and scope. It is also worth noting that niacin's slower, steadier elevation of NAD through the Price-Handler pathway may confer benefits that avoid the pitfalls of acute NAD spikes. Excessive NAD elevation through salvage pathway precursors like NR or NMN could potentially lead to an overproduction of nicotinamide, NEM, a feedback inhibitor of sirtuins, unless it is efficiently cleared via methylation. In high doses, this can strain the methylation cycle and paradoxically blunt the very longevity pathways they aim to stimulate. Niacin, which bypasses the salvage pathway's early steps and produces less immediate NEM, may offer a more balanced and sustainable route to NAD restoration. Moreover, niacin's clinical utility in age-related diseases is not hypothetical. It has been formally studied in atherosclerosis, age-related macular degeneration, Alzheimer's disease models, and certain neurodegenerative conditions. Its pleiotropic effects, including enhancement of blood flow, reduction of oxidative stress, and stabilization of mitochondrial function, are not merely theoretical extrapolations from animal data. They are part of an extensive clinical record that spans decades. While it is true that niacin's use in cardiovascular disease prevention has become less prominent in modern guidelines, this is largely due to concerns about side effects in statin-treated patients and shifting priorities in lipid management, not because niacin is ineffective. On the contrary, its metabolic benefits remain relevant, particularly for individuals who are statin intolerant or have broader metabolic syndrome. In the context of aging, where systemic resilience, mitochondrial capacity, and inflammatory regulation all play central roles, niacin's proven impact on these domains deserves renewed attention. Its affordability, accessibility, and well-established safety profile, especially in low to moderate doses, make it a uniquely pragmatic candidate in the search for viable longevity interventions. In conclusion, while NR and NMN represent exciting and promising additions to the NAD boosting toolkit, their clinical utility remains in the early stages of validation. Much of their anti-aging allure is rooted in theoretical mechanisms and animal studies rather than established human outcomes. Niacin, by contrast, is a compound with a long clinical track record of improving the very systems that deteriorate with age: lipid metabolism, vascular integrity, mitochondrial function, and inflammation. Far from outdated, niacin may offer a more scientifically grounded and therapeutically realistic path toward healthy aging than its high-tech counterparts. Its reputation may benefit from a fresh reevaluation, not as a lesser alternative, but as a legitimate, clinically validated, and metabolically intelligent anti-aging intervention.